And what it is basically on the y-axis over here, you're going to have, this is the percent of oxygen saturation, the percent of O2 saturation. Okay, so they're percentages. This is 50%, here's 25%, here's 75%, and this would be a whopping 100%. Okay? On the axis over here, this will be partial pressure of oxygen. All right? So the amount of oxygen. And this will be in millimeters of mercury. Okay, pressure. And for our purposes, you could also visualize it as like percent, how much of the oxygen you're actually breathing, percentage of oxygen. But it's really under millimeters of mercury. And again, I'll put it where it's 50, 75, 25, and here's 100. All right. Again, 25 millimeters of mercury to 100 millimeters of mercury. But you could also try and, if you want to visualize percentage, just know it's not percentage, it's really millimeters of mercury. So what's happening here is that you would, if there was 25 millimeters of mercury here, then you would expect 25% to be filled. If we say there's 50 millimeters of mercury of oxygen coming in, 50%. But if that happened, it would be a linear line like this. But this oxygen dissociation curve is not linear. What's happening here, in order for us to get this to be, let's say, 25%, we have to put more pressure of oxygen to get that to 25%. So in other words, it's not going to be 25 to get 25. We might have to, I don't know the exact numbers, but let's say we might have to bring it up to, let's say, 30 over here. Now this will actually bring it up to 25%. So we're putting more pressure over here, oxygen, more oxygen pressure to get to 25%, to get that first oxygen on hemoglobin. Remember, there's 4 oxygens that go on hemoglobin. The first one is very hard to get on. You're going to have to put an extra effort into it, so to say, an extra amount of pressure. So here we're going to get our first one over here. But then, once that one goes on, the second one happens much faster. This one actually took the trouble to use the extra effort, but it's helping the second one and even the third one to go on much more rapidly. So we don't need to actually go up to, so here's the second one, here's the third one. We don't need that much pressure to get the second and third one on. Does that make sense? The first one we do. And then the last one actually doesn't really need that much pressure over here. Actually, this, oh no, I'm sorry, I did this wrong, hold on. So that's, yeah, that's right, I did it. So it's 25, 30, or 75%, and then let's say this will be 100 over here. So this is the line that you're going to be getting, is more of like an S shape. Does that make sense? Again, my little scenario of this, which you could actually visualize it, is take a 4-block stamps. If you got stamps, like postage stamps, in order to take the first one off, we have to put more effort into it. We're going to have to break off this bond and break off this bond for us to take the first one off. There's more effort to do that. But because that first one took off the 2 bonds over here, it's going to make the next 2 much easier to come off. All you have to do is just break one bond for this one, and this one comes right off. You only need to break off that one bond, and then this one comes off. And this one is very easy to come right off, it's all by itself. Does that make sense? It's what we call cooperative binding. That the first one is going to cooperate to make the other 2, 3 to come off much faster. Is that clear about that? Okay? So that's what this is. All right? It's called the oxygen dissociation curve. It's not a straight line. It's more or less like an S. It's kind of shaped really like that, though. Okay? Now, there's different factors that are going to play here to do something different to this curve. That's what we got to talk about. All these different factors. These factors include the partial pressure of oxygen, as we just saw how the more oxygen, you can see what's happening, more oxygen is going to be able to get more oxygen onto it, right? So that changes things. I'm not going to go into that one. But the amount of carbon dioxide in the blood is also going to change, right? It's always going to help alter this curve, and I'll show you that. So the partial pressure of carbon dioxide, also the blood pH is going to do something. Whether your blood is more acidic or more basic, it's going to do something here. And remember, blood pH has to do with the amount of hydrogens. Going back to chemistry, so we're going back to chemistry with this. Also, temperature is going to play a factor in this to do something to this curve. We're also going to talk about something called the concentration of ppg, also known as dpg. And we'll get into that later. It's an organic chemical that's it's found on red blood cells. We'll talk about that. And also fetal hemoglobin is also going to take part in this also, because we know that when a fetus inside the mom, the lungs are not working. So when the oxygen comes onto the hemoglobin of the fetus, we've got to make sure that it's going to stick a little more tighter than usual in an adult, because there is no interaction with the lungs. We've got to have it stay on a little bit longer. So any change in these both factors, whether we increase or decrease the temperature, increase, let's say, the partial pressure of carbon dioxide, it's going to alter this whole curve. It's either going to make this curve shift to the right or shift to the left. So we've got to talk about what that is. A shift to the right means that the hemoglobin needs more affinity, more attraction. Affinity means traction, like two magnets. They have a strong affinity with each other, strong attraction. So if there's a shift to the right, the hemoglobin needs more of an affinity, more of an attraction to bind to O2. It's a very weak magnet, in other words. It needs a stronger magnet. Otherwise, this oxygen is going to come right off. Does that make sense? So if there's what we call a shift to the right, which kind of will look like this in purple, it'll look like that. And what that means is that we're going to need more of an affinity for oxygen to go on there. Otherwise, if we have a shift to the right, oxygen is going to come off, because it's a weak magnet. Does that make sense? Okay? So if there's a shift to the right, oxygen wants to get released from the hemoglobin. It's much easier for the oxygen to fall off the hemoglobin if there's a shift to the right. Then we also have a shift to the left. And a shift to the left, which I'm doing in green here, will look more or less like that. It's going to move over to the left side. Now this doesn't need much of an affinity to bind there. These are already strong magnets. Oxygen is going to be really bound hard to this. Look at this. For us, normally, in the red here, we need a lot more pressure to get a lot more partial pressure of oxygen to get it to 25%. But over here in the green, we don't need that much because it's already a strong attraction. If I show you the purple here, you need a lot more pressure to get it to 25%. So it needs a stronger attraction. If you want it to stay on there, you're going to need a lot more attraction. So the green here, a shift to the left means that oxygen wants to stay bound to hemoglobin. Is that clear about that? Okay? And these things that I'm telling you about, pH temperature is going to either make it a shift to the right or shift to the left. If it's a shift to the right, oxygen tends to come off because it has a less of affinity towards it. If there's a shift to the left, then oxygen is going to be staying bound to it. You're going to have to do something to try and get that hemoglobin or that oxygen off. Okay? My little mnemonic for this is to think of the Wright brothers, you know, the first men flying on planes. They have that weird kind of airplane, you know what I mean, where they're lying down. You've seen those things, right? Well, think of them, the Wright brothers, Wright brothers are dropping oxygen tanks off their planes. They're just dropping them off. So a shift to the right means that you're going to release quite easily a lot of oxygen. Okay? Don't memorize anything to the left. Make your life easier. If you know the right means you're going to release oxygen, then the left means it's going to stay on. You don't have to memorize it. Don't waste your time on that. Okay? All right. Are we clear with that? Because we're going to start getting heavier now into these factors and how they actually do this. Okay. So the dark line up here is the normal person, so to say. If we're going to make a shift to the right, now shown in dotted line up here, what causes a shift to the right, which means that more oxygen is going to come off, it's going to be easily released. It has a low affinity for onto hemoglobin, so it comes off quite rapidly. If we increase the PCO2, the partial pressure of carbon dioxide, lower the pH, increase the temperature, or increase the DPG, which we'll talk about. If there's a shift to the left in this graph, so the solid line is the normal, but now the dotted line is showing a shift to the left, this is when we have a decrease in partial pressure of carbon dioxide. When the pH goes up, and let me just ask you, let's just see where you are with this chemistry thing. If the pH goes up, does that mean we're losing, if the pH goes up, does that mean there's more hydrogens in our solution or less hydrogens? Less hydrogens, right? If your pH goes down, it's very acidic, which means there's a lot of hydrogens there. If your pH goes up, it means that your hydrogens are going to be less, right? Alkaline and acidity. So again, if your partial pressure of carbon dioxide goes down, your pH goes up, or your hydrogens go down, your temperature goes down, or we have a decrease in this DPG, or if we're talking about hemoglobin, that's fetal hemoglobin, that's what that is, hemoglobin F, that's fetal hemoglobin. Then there's going to be a shift to the left. Okay? I'm just showing you as an overview, because we're going to go into step by step about things. Okay? Is that clear? Shift to the right, shift to the left. Expect questions, a lot of them, to be put on the next exam. Okay? But they're also in the review questions, a lot of them. So you should be able to manage the test if you do those questions correctly. Okay, so influencing. Let's talk about the partial pressure of carbon dioxide. We already did the partial pressure of oxygen. I showed you how that affects this curve. Okay, with the left to right. So now let's talk about the partial pressure of carbon dioxide. Carbon dioxide, let me give you a little background. Carbon dioxide actually is in the body in three forms. All right, once it gets into your body, 7 to 10% of it will float in the plasma of your blood as carbon dioxide gas. Okay, it gets dissolved in there. It's pretty easily dissolved when you compare it to oxygen. So about 7 to 10% gets dissolved in the plasma. We also have some that actually are bound to hemoglobin. About 20% of the carbon dioxide in your body is put onto and bound to hemoglobin, where oxygen usually is. Okay? 98% of oxygen is on hemoglobin. Well, carbon dioxide could go on there too. Not so great, still about 20% though goes on there. But the whopping amount of carbon dioxide that's in your body gets converted to bicarbonate ions, right? Remember the whole reaction that we did. The bicarbonate ions is where the CO2 is. So let me show you this equation again. right? You know this reaction? I guarantee it will be on exams. Trust me. You're going to have to redo that. If there was a if there's a tattoo that you want, that's probably the tattoo you want to get. All right? It's gonna be one of these. I keep on telling people that, and it's going to be one of these days someone's going to have a tattoo. Don't. I'm not encouraging tattoos. I'm just emphasizing that this is a very important reaction. You've seen it in the parietal cells of the stomach. You've seen it now. You should have saw it up in the brain in AMP1, okay? But you're going to see it more often. You'll see it in the pancreas as you saw before and stuff. So this whole reaction, very important. All right? So what happens here is that carbon dioxide, 70% of carbon dioxide in your body is in this form right here. Bicarb. I call it bicarb, but it's bicarbonate ions, which is HCO3 with negative charge. Okay? That's where it mostly is. Hint, hint, you should know that. Okay? Now, students think that hemoglobin transports most of the carbon dioxide. That's wrong. It transports most of the oxygen. But most of the carbon dioxide is in the form of bicarbonate ions. Okay? So this is just showing you in a picture form. You know, here's the carbon dioxide that comes in. 7 to 10% is dissolved in the plasma. The other 93%, you've got about 20% that binds to hemoglobin. The other 70% gets converted and goes back out into the bloodstream as bicarbonate ions. So yes, this reaction, which is actually catalyzed by carbonic anhydrase, right over here, and it could go both ways, right? You see now the video where the reaction is reversible. It can go both ways. That's where the carbonic anhydrase occurs. So, carbon dioxide diffuses into the red blood cells and combines with water. CO2 combines with water, immediately turns into this carbonic acid that's only intermediate, that stays there for a very short time. We're talking about milliseconds. Milliseconds. And eventually will quickly actually turn into hydrogen ions and bicarbonate ions. Okay? So therefore, our red blood cells, where this occurs, contains carbonic anhydrase, and it reversibly does it. Okay? So that reaction is the same thing up here. Okay? Now, at the tissue level, not the alveolar level, at the tissue level, you have bicarbonate ions that are down in the bloodstream down there, and the bicarbonate ions need to change back to carbon dioxide to get into the tissues. So they could quickly diffuse into the red blood cell where it meets up with carbonic anhydrase. The carbonic anhydrase is going to mix the bicarb that just entered. There's also hydrogen that's in there, and it's going to go this way. Now there's something called the chloride shift. The chloride shift is that in a red blood cell, if you're going to put bicarb into the red blood cell, are you making it more negative inside or more positive inside? Come on, guys. Bicarbonate ion, what charge does that have? Negative. So if you get a whole potload of these bicarb ions going inside the red blood cell, is it going to make it more negative, more positive? Negative. Okay? But we don't like that. So what happens here, in exchange for that new negative charge coming in, chloride, which has a negative charge, is going to go out to keep the charge equal. Homeostasis. We call that the chloride shift. So this keeps the electrical balance maintained. So this is what's happening at the tissue level. All right, don't worry about this at the moment. Here's a red blood cell in the bloodstream. Bicarb is going, tissue. Bicarb is going in here. Oh, okay, I'm sorry. So what's going to happen is bicarb is going to go out here. Chloride will go back in. You see how it's like the bicarb has a negative charge and chloride has a negative charge? If one's going out, one's going in, it's going to keep the charge equal inside. Does that make sense? Yes? Okay?
If I put the potassium and the potassium goes out
For aldosterone. But yeah, that's what happens with that. So what's happening here, look at the tissues. Let me show you again. Here's a tissue, carbon dioxide, because we're having cellular respiration happening. It's going to make ATP. Carbon dioxide comes out of the cells over here from the mitochondria, goes out of the cells and goes into the bloodstream. The carbon dioxide, only 30% of it's going to stay in the plasma, but 70% of it, 20% is going to stay in the plasma, 10% stays on the hemoglobin, and 30% of it needs to be changed into bicarb ions. So what's going to happen here is the carbon dioxide will go diffuse into the red blood cell. You have carbonic anhydrase in there. The carbon dioxide mixes with the water, changes into carbonic acid, and immediately will change into bicarb ions and hydrogen ions. Just the reaction I just put right up there. Now the bicarb can come out into the bloodstream, but chloride needs to go into the cell to keep that negativity, or not negativity, but to keep the charge the same. That's what we call the chloride shift. Does that make sense? Now, most of the carbon dioxide is in the form of this bicarb, and it's floating in your bloodstream as an ion. Okay? Is that clear about that? Now, let's look at the lungs. You're going to breathe in carbon dioxide. Okay? So, you're going to breathe out carbon dioxide. So it's basically the reverse of what we just talked about. Bicarb ions you can't breathe out ions. It doesn't happen. You got to change it back to a gas. So we got to change the ion into a gas. So the only way to do that is to use carbonic anhydrase again. So this bicarb ion is floating around into your bloodstream, into your plasma, and that needs to be converted. So what will happen here is that it's going to, here I'll just show you the picture, right? Carbon dioxide fuses. So here's at the lung level. So here you have bicarb that reached the lung level, but you can't blow out bicarb into your lungs. This is your lungs over here, alveolus. But you can't blow that out. So you have to turn this back into a gas. So the bicarb ion goes in a red blood cell. That whole reaction occurs again, carbonic anhydrase. But if we're going to put a bicarb in here, that's negative. You're going to make inside more negative. So we have to get rid of a negative. So we have the chloride shift happening again. As bicarb goes in, chloride goes out. Does that make sense? Okay. Does the whole reaction, turns it into carbon dioxide gas. The carbon dioxide gas at the level of the lungs is able to pass right through here through simple diffusion, and it goes into the alveolus, and you can breathe out carbon dioxide gas. Okay? Does that make sense with that? Do make sure you know that reaction. Hint, 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 hint. Okay? There's the reaction again. So the amount of carbon dioxide transport is markedly affected by the amount of O2. Okay? There's an effect called the Haldane Effect. And what it is that low levels of oxygen that's bound to hemoglobin, this enables blood to transport more carbon dioxide. All right? It's a phenomenon that happens. So oxygenated hemoglobin does not bind CO2 as well as deoxy. In other words, if you have a full amount of 4 oxygens on one hemoglobin, it's hard to get carbon dioxide on there, so the carbon dioxide can't be bound to it. That's what it's saying here. Okay? If there's only 2 oxygens on there, then carbon dioxide can bound to it. Okay? So deoxygenated hemoglobin, which is that HHB, binds more hydrogen than this. So as hydrogens are removed, this reaction moves a certain way. If we we remove the hydrogens over here, does the reaction want to go this way? Or does the reaction want to go this way? You're going to have to do this understanding. It's called Le Chatelier's principle. It's going to want to go this way. Think about it. If there's reactants over here, and you're going to remove some of these over here, these products over here, we have to keep a balance. As you've seen in the video, there's got to be a balance of the two. If we have, on this reaction here, we have 50% of the products here and 50% of the products over here. That's balance. It's a teeter-totter. And you can do that because the yield signs here go both ways. So you could do this. In other words, if we're going to lose some of these hydrogen ions, as I'm showing you over here, then technically what's happening here is that you're going to have maybe 30% of products here because you're missing some of these. Net-wise, you're missing some of these. So therefore, compared from this side to this side, there's going to be 70 here. So if there's less over here, which way does the reaction want to go to get it back to 50-50? Does the reaction want to go to the door, or does it want to go to the wall? It wants to go this way, so that these products here will go down to, let's say, 50, making this over here a little bit more to get to 50. That's how this works. So Le Chatelier's principle. Okay? That's what I'm trying to show you up there. Okay. If you increase the CO2, you're going to decrease the pH. Think about it. If you increase the CO2, and you can't blow it off, which way does the reaction want to go? If you increase the CO2, normally you could blow this off, but for some reason, you can't. If you're making too much of it, you can't blow it off that fast. Then which way does the reaction want to go? That way, right? If it goes this way, are you making more hydrogens or less? Let me finish this and I'll do it. If your CO2 goes up in your blood, you can't blow it off, then which way does it go? It goes this way. When it does, you're going to make more hydrogen ions, are you not? When you make more hydrogen ions, what happens to your pH? It goes down. So that's what I'm saying here. If you increase the partial pressure CO2 in your blood, you're going to increase your hydrogens, but that will decrease your pH, make it more acidic. Is that clear about that? Question? Yeah? It's like an epiphany. Once you get this, it's like, why did I have a hard time in the first place? It will make sense to you. You can do either way. When we get into fluids, electrolytes, and acid base, a whole chapter about this more, you will, and that's why I'm putting this respiratory with fluids and electrolytes with your urology. I know it sounds like 3 chapters a lot, but they go together. So you'll be seeing this on urology, you'll see it with fluids and electrolytes. You'll hopefully get it by then. Okay? Questions about that? All right? There's the reaction. I'm putting it up there so many times. I don't know how else to emphasize. You need to know the reaction. Okay? So acid-base balance through respiration, right? Normal blood pH is what it is between 7.35 to 7.45. So we're looking at 7.4 is the blood pH. It's a very weak alkaline environment. The distilled water is 7.0, so it's this. If it's less than 7.35, I guarantee you'll be sick. It's just a small window, okay? So this is what we call a buffer system. If we put more hydrogens in here, it's going to get rid of them by blowing it off. If you're going to decrease it, it's going to make more. It's a buffer to keep it 50-50. That's what a buffer system is, so that the pH remains the same. Okay? So if you increase the hydrogen ions, you're going to have a lot more hydrogens in your bloodstream. That's going to be for more of a acidic environment. So if you make too much hydrogens, it's going to combine it with the bicarb over here, and guess what organ gets rid of ions? If carbon dioxide, you get rid of that by way of the lungs, it's gas. How do you think you're going to get rid of these water-soluble things? What organ? Kidneys. So this is where the kidneys and the lungs work together. Okay? If you decrease the ions of hydrogen in the blood, then the carbonic acid is going to dissociate and release more hydrogens. So that's how carbon dioxide affects this. Now, let's talk about pH, which we've kind of done already, or leading to it. There's something called the Bohr Effect. It's a phenomenon that happens. We can't really explain it. It just is going to happen. That when you have low pH and an acidic environment, this weakens the hemoglobin and oxygen just wants to come off. You're going to have a shift to the right. Oxygen wants to come off like the Wright brothers. They release the oxygen. You have a shift to the right when your pH goes down, right? An acidosis kind of environment, or acidotic. We call it the Bohr Effect. So this accelerates the unloading of oxygen when you have your environment more acidic. All right? So a decrease of pH, which means an increase of hydrogen, will be a shift to the right, meaning that there's going to be more oxygen coming off the hemoglobin. All right? This kind of makes sense. Think about this. You're running a marathon. Right? You're running a marathon, and then all of a sudden you get this pain in your side. Ah! What is that pain that you feel after 2 or 3 miles? What is that you're building up in an anaerobic environment that you're doing? What is it? No. Lactic acid. Acid. So you're actually building up acid in your bloodstream because you've used up all the oxygen that you needed, so your body knows how to make ATP. Not very efficiently, but it knows how to make ATP without oxygen. Anaerobic respiration. The byproduct of that is lactic acid, which is what you feel, and it causes fatigue. You get tired from it. Okay? So if you build up lactic acid when you're running a marathon, your hydrogens in your body go up. When they go up, does the reaction want to go to the wall or to the door? To the wall. Does that make sense? We're putting more over here, right? So this way is, if we're using the numbers over here, these arbitrary numbers, this would be 70% and this would be 30%. So if this is going to go up, the reaction is going to go this way. Are you making more carbon dioxide or less? More. And when you make more CO2, if you're running a marathon, are you hyperventilating or are you hypoventilating? Are you breathing very fast or are you breathing very slow? Fast. Because you're making a whole whopping amount of carbon dioxide to get that out of your body. That's what you're doing. All the lactic acid that you're building up. Is that clear with that? Okay? So the Bohr effect is that oxygen wants to get off hemoglobin, but the Haldane effect is carbon dioxide wants to get into the hemoglobin. So as CO2 enters the blood, it causes more oxygen to unload. That's the Bohr effect, which in turn allows more carbon dioxide to combine with the hemoglobin and more bicarb is formed. That's the Haldane effect. You got to understand the Bohr effect and the Haldane effect. All right, the phenomenons that happen, but you need to know those words. Okay? Let me do temperature. Because it's pretty easy to understand this one too. If you increase the temperature, the hemoglobin will allow the oxygen to come off quite easily, i.e., a shift to the right. The hotter you get, the temperature that you get, the more likely oxygen is going to get released off your hemoglobin. Think about it. You're running a marathon. Are you building up more temperature, or are you losing temperature? Or that doesn't sound right. Are you getting hotter, or are you getting colder? Hotter, right? We all do that. So that's another mechanism that's going to make a shift to the right. You produce more energy, you're going to get hotter, and that's going to allow the oxygen to come off to go to the tissues. Because that's where it's needed. Because you're getting really hot. Now, you know how I start asking questions? Because I want you to make sure you know endocrinology also, which is the first chapter of this course. In a person who's got hyperthyroidism, are they increasing their metabolism or decreasing their metabolism? Graves' disease. Increasing their metabolism. Are they producing more ATP or less? More. Are they getting hotter or colder? Hotter. So in people who have hyperthyroidism, they're going to have a shift to the right. More oxygen's coming off. People with hypothyroidism, someone who's maybe also in a freezer and freezing there, they will have a shift to the left. That oxygen doesn't want to come off. You're not doing any kind of exerting energy. You're not getting hotter in there. And that's what's happening. Okay? So, if you increase your metabolism, like in hyperthyroidism, or anything, you're running, you're going to increase your temperature, oxygen wants to get released. It just comes off. It has a lower affinity for oxygen, so it just comes right off. They get weaker magnets. It comes off. Okay? So here, this is just showing you the pH and temperature, shift to the right, shift to the left. This is the pH, and this is stuff I've been talking about. And here's the temperature, how that changes the oxygen dissociation curve.